All right, let's talk about mitosis now, each of the steps of mitosis. We're going to start with interphase. And I know interphase is not part of mitosis, it's not part of the M phase, but we have to talk about specific parts of interphase that to be able to transition into cell division. So one thing that happens during interphase is going to be the S phase, right? S phase is a subphase of interphase. And what happens here during S phase, we get DNA duplication. In other words, we make a copy of DNA. And this is important because if a cell is supposed to divide into two cells, each cell must have the same amount of DNA. Okay. During late G2, two centrosomes are going to appear. Now, let me define a centrosome. A centrosome is a microtubule organizing center. It is made of or contains two centrioles. These are going to be very important because they're going to be producing something called spindle fibers. And we'll see them in a little bit. Okay. And the spindle fibers are going to help, they're going to guide the chromosomes move to opposite directions. So here are going to be our centrosomes, and I'm going to number them that way you can follow me. Centrosomes over here. And each centrosome, now they're duplicated, they're going to be moving to opposite poles of the cell. Now, by this time, in interphase at the very end, late G2, the chromosomes have been duplicated, right? We have already made DNA. We have gone through S phase. However, they're still uncondensed. They're loosely packed. And that's what you see over here. So I'm going to put here number 2. Okay, still uncondensed. In other words, what you see over here, okay, which are is pointing over here. What you see here is spaghetti, right? You really cannot tell the different chromosomes or tell them apart. Then we continue with prophase. And in prophase, the chromosomes are now condensed, right? Now we have this chromosome type of the DNA is now in the chromosome shape, right? So if I can, I'm gonna try to do a little bit of a art here for you guys. This is what a chromosome kind of looks like now, and this is gonna be the centromere. Okay, this is now what a condensed chromosome kind of looks like. Each chromosome is gonna consist of two identical sister chromatids. So two sister chromatids. This is one, and this is the other one. Think of them as arms and legs. Okay, so I'm going to put that over here. Number 1. I'm going to put this as number 2. Okay, number 2. Please don't get confused. This is number 2 for sister chromatids. And I put here number 1 as this condensed chromosome. Now the centrosomes have moved toward opposite poles, like I told you they were going to do. So this is going to be 3, and here are our centrosomes. Centrosomes over here, okay, they have been moving to opposite poles. So that's going to be number 3. Number 3. Mitotic spindle starts to form. The mitotic spindle, these are going to be kind of ropes of something called microtubules. Okay, and that's gonna start to form. And then mitotic spindle, also known as spindle tracts, they're gonna be over here. Okay, and they're pointing to these things over here. All right, all of this kind of rope type of thing here that are gonna guide the chromosomes into moving from one place to another. The nuclear envelope breaks apart. That happens during prophase. Okay, we can see here fragments of the nuclear envelope. In other words, the nuclear envelope has broken apart. And the spindle tracks, again, the spindle tracks are made of microtubules. I'm going to attach the centromere of each chromosome and move the chromosomes towards the center of the cell. Okay, and that's what's happening in this picture over here. You see, if you can tell this is going to be attaching to the chromosome over here. This part here in the middle, like I said in my picture, this is going to be a centromere. The one in the middle, that's going to be a centromere. And remember, how do you count chromosomes? How to count chromosome number? Count the centromeres. Count the centromeres. Okay, so if I go to my picture over here, I can count 1, 2, 3, 4, 4 centromeres. I see here 4 chromosomes. Remember from my previous videos, even though you see chromosomes duplicated, right, as long as they're attached, as long as the sister chromatids are attached, okay, I'm gonna actually draw this here. This part here, that's a centromere as well. As long as they're both attached, both sister chromatids are together, it is still considered one chromosome. And after prophase, we go to metaphase. And in metaphase, the spindle, mitotic spindle, is going to be fully formed. Okay, so I'm going to put here number 1, and this is going to be like number 1. You see how all of these ropes are here attaching to some of them attach to the chromosomes. Some of them are going to be attaching to other microtubules. Okay, so whenever you hear spindle, mitotic spindle, again, these are a bunch of microtubules. The chromosomes are not going to be at the center. Chromosomes at the center over here, they're all in a line. Okay, and that was a terrible line. Let me attempt this one more time. There you go. Hopefully you get to see the line. Okay, and then, by the way, how do we keep everything together? Mitosis, metaphase, right? Metaphase, M for middle. If you remember that, you'll remember that if it's metaphase, it starts with M, that means middle, that means all the chromosomes are at the middle, okay, at the center of the cell. Uh, and then the sister chromatids, okay, are attached to a spindle fiber at the centromere. And that's what you see over here. Number 3. You see how all of these things here, let me be, how this, for example, this one is attached, this spindle fiber is attached to the centromere of a chromosome. And then we have anaphase. And during anaphase, sister chromatids separate. And this is the key part. Okay. The chromosomes, they have sister chromatids, they're finally going to separate. And each chromatid is now going to be considered a full-fledged chromosome. Okay, not just a not a sister chromatid anymore. Now it is a chromosome, no longer called a sister chromatid. Okay, so I'm gonna put a note over here: sister chromatids only when attached to centromere. If they're separated, they're no longer chromatids. Now we have chromosomes. The chromosomes move toward opposite poles, and that's what's happening here. So I'm gonna put I'm gonna make this number 1. Okay, Number one, as you can see, the chromosomes now are going to be moving to opposite poles. For that to happen, the spindle tracks or the microtubules. Okay, so you don't get confused, think of the spindle tracks as something made of microtubules. So think about the spindle tracks as a team and the microtubules as the different players in that team. Okay, so some spindle tracks are going to shorten, and these are going to be attached to the chromosome. So for example, this one here is going to shorten. This microtubule is going to shorten. But there's going to be other spindle tracks that are not attached to the chromosomes, that are going to push the poles further apart. Okay, and that's going to be, for example, this one over here. Okay, I'm overlapping it, but you see how they're kind of microtubule to microtubule here, and they are going to be doing the opposite, going to be pushing each other away. That way the cell can split, and then this is going to elongate the cell. Now, in anaphase, like I said, this is the key thing, all right? Sister chromatids separate. So another way to understand this or to keep it straight, in this case, anaphase starts with A. Think of A as a way. And if you keep that straight, I mean, it's really hard to screw this up, all right? You kind of already know the stages of mitosis, right? Metaphase in the middle, all chromosomes in the middle. A anaphase, A away, chromosomes are separating, they're moving away from each other. And then we have telophase. And in telophase is going to be the reverse of prophase. So whatever happened in prophase is going to be the opposite of prophase. So reverse of prophase, or I'm going to say opposite of prophase. Reverse of prophase, or I'm going to say opposite of prophase. Chromosomes are going to arrive at opposite poles. The nuclear envelope is going to form. I'll put here number one. Number one, as you can see, little tiny little bits here are kind of coming together. The nucleus is coming together. The chromosomes are going to uncoil. They decondense. I'm going to put this as number two. I'm going to put this as number two over here. As you can see, now the chromosomes are not like they don't have like two legs and two arms, right? Now they're very much decondensed. They start looking like spaghetti. The spindle is going to break up. All right, the spindle fibers breaks up. I'm gonna put that. I don't think you cannot really see it here, but let's just put it over here, okay? As saying as the microtubules are breaking apart. Two nuclei appear, and at this time, while in the last part of mitosis, we start to seeing the beginning of cytokinesis, okay? And cytokinesis, we'll go into more detail in a second, but cytokinesis is gonna be the division of the cytoplasm. And whatever the components of the cytoplasm are. For example, organelles and that kind of stuff. Okay, so that is what happens during metaphase. Now you know what happens in prophase, metaphase, anaphase, and telophase. And an easy way to remember it is remember what prophase looks like, and telophase is going to be the same but the opposite. Right? Metaphase starts with an M, that means the middle. That means all chromosomes are in the middle. And anaphase starts with an A for away. Chromosomes are moving away.